Good morning, good afternoon, and just to be sure, good evening, everyone. Welcome to the 14th Pan-Asia Farmers Exchange Program. This is day 2 of the 5-day webinar that we will be holding until the 20th of November. I am your moderator for today. I am Abe, Abraham Manalo, Executive Secretary of the Biotechnology Coalition of the Philippines. And I would like to thank my co-organizers for this Farmers Exchange Program 2020 with CropLife Asia, here with the beautiful Sanita Baba, and the dashing Executive Director of CropLife Philippines, Mr. Edilberto De Luna. I would like to welcome all participants, especially our friends who are joining from outside the Philippines. So please let me mention the 13 economies that will be and have joined us yesterday and today, and most probably during the next 3 days of the Farmers Exchange Program. And I would like to welcome, of course, from my beautiful country, the Philippines, and the other 12 economies, Taiwan, Korea, Pakistan, Thailand, China, Indonesia, Singapore, Australia, Japan, Vietnam, India, and Malaysia. So thank you for your participation, our delegates from the 13 economies. And I'm seeing here some people who are saying hi to everyone. So hi also to everyone out there. So before I introduce the first speaker, let me give you some points to remember for the duration of the program. Number 1, please, if you have questions, type these at the Q&A section anytime during the presentation or even during the Q&A portion. And I will be reading your questions to the presenter who just gave the lecture. Number 2, questions will be answered at the end of the presentation. If there are questions that cannot be accommodated during the session due to time constraints, this will be answered through the chat box by the presenter in charge or via email. So our secretariat will be pulling all questions that have not been answered through the chat box or live, and they will be answered at least via email. Number 3, e-certificates will be given only to participants who will be able to attend the 5-day program. These certificates will be sent to your registered email addresses. And number 4, every day, including most especially today, there will be a $20 question at the end of the session. So we will have one question to be answered by all participants, and the prize is, we will have 3 prize winners, and each prize winner will be getting a voucher equivalent to, drum roll, $20. So let's get this webinar going. Our first speaker for today's session is considered one of the pillars of Philippine biotechnology. And I'm very, very close to this lady, to this great lady. She has helped craft biosafety policies in the Philippines, including the first national biosafety policy, the policy on GM crop regulation and related issuances, and the present National Biosafety Policy in the Philippines, which is Executive Order 514, Series of 2006. She has established the first functional forensic DNA laboratory in the country, which is instrumental in the acceptance of DNA evidence by the Supreme Court in 2001. So if you have problems with paternity, you have to we have to thank Dr. Nina for that. She is presently the president and chair of the board of directors of BCP. She also serves as consultant in capacity building and biotech regulations to the Biotech Program Office, the Department of Agriculture, and a technical advisor to the Climate Resilient Agriculture Office of the also of the Department of Agriculture. She graduated magna cum laude for her BS Agriculture degree from UP Los Baños, and she holds a PhD in genetics from the University of California, Berkeley. Today, she will give us an overview of biosafety regulations, including the environmental risk assessment and food and feed safety assessment of GM crops. Ladies and gentlemen, please let us all welcome Dr. Saturnina de la Cruz Halos. Ma'am Nina.
Thank you, Abe. Okay. Yeah, let me have let me share my screen there. I have to. There. Okay. Okay. Hello, everyone. I hope you're okay. There's still COVID going around, so you better take care and follow all the health protocols. We do have them in the Philippines, and I hope you do have them in your countries too. So we'll talk about biosafety regulations, just a general view of what biosafety regulation is. What are the common issues that is addressed by biosafety regulation, which would be food safety and environmental safety. And we'll talk about how food safety is assessed, as well as how environmental safety is assessed. Oops, what happened there? Okay, biosafety regulation refers to a policy that is issued by the government to protect human health and animal health and the environment from the possible adverse effects of products of modern biotechnology. And this policy would define the system of regulation. The products of modern biotechnology are referred to as GMOs, LMOs, biotech crops, transgenic organism, recombinant organism also. It's not listed in here, but it's also one of the terms that are used for the products of modern biotechnology. And then genetically engineered organisms. These products of modern biotechnology are developed in the laboratory, and they use techniques that would directly alter or change the genetic material. And our genetic material is DNA. That's for more advanced organism. Some viruses have RNA as their genetic material. Or the product of modern biotechnology could also be obtained by fusing cells of unrelated organisms. And this is, obtained from, this is just a rehearse of the definition given by the Cartagena Protocol on Biosafety on the products of modern biotechnology. The regulatory systems would differ from country to country. There are some countries that drafted a particular law specific to biosafety regulation. For example, Brazil, or even Australia. On the other hand, there are countries that derive their regulatory system by looking or getting the provisions of existing laws. Like in the Philippines, we have a biosafety regulation that is based on existing laws that covers food safety and environmental safety. And the coverage of this of the biosafety regulation, again, this this would differ from country to country. The definition, for example, in other countries would refer to products of modern biotechnology, and that means that this would regulate not only LMOs, GMOs, but also organisms that have been altered genetically, equivalent to a mutated organism, or what we refer today as the genome-edited organisms. On the other hand, and this is under the Cartagena Protocol on Biosafety, the regulation would cover only the products of modern biotechnology with new genetic combination, which is your LMO under the Cartagena Protocol on Biosafety. And this would exclude, or it would not cover, products of genome editing. This is the regulation that we have today in the Philippines, that we have a coverage only for products of modern biotechnology with new genetic combinations. Now, the basis of decision could be based on the results of a risk assessment. We're looking at the possible dangers in terms of food, or when it is ingested in humans, in terms of feed safety, environmental safety. There are other countries that would consider social, ethical, and economic impact of the particular product. Today in the Philippines, we have food safety, environmental safety assessments, and we also consider the social, ethical, and economic aspects of the product. The common issues among countries is to look at the possible dangers in terms of food and environmental safety of biotech products. So it's safety as food and feed, and safety for the environment are the common issues that are addressed by biosafety regulation with GMOs. Now, when it comes to food, the focus for evaluating whether the food derived from a GMO would be usually focused on the protein product of the transferred gene. And this is because that's the new thing that is present in that GMO. Usually the GMO is compared with the non-GMO counterpart, and it is often that the new protein product is the difference between those two, so that the safety of that new protein product is the one that is assessed. On the other hand, because genetic engineering would make some changes in the DNA, like, for example, it would insert the new gene into the genetic material, then this could also change the nutritional or natural hazardous components of the food. So these are the focus of the risk assessment or food safety assessment for GMOs. Now, why are we assessing only the protein product, since you are transferring a gene? The proteins are usually, they can cause some problems like allergenicity or toxicity. On the other hand, the transferred gene has the same composition as the genetic material of the host organism or the non-GMO itself. So it is not a safety issue. On the other hand, there are possible changes in the nutritional and natural hazardous components. There could be a decrease in key nutrients. There could be an increase of the native toxin, allergen, or anti-nutrient in the host organism. Well, food, we ingest it so that it would contribute to our well-being and health. However, we have known hazards that are associated with food. And the major ones are allergenicity, toxicity, and anti-nutrients. Allergenicity is the potential to cause allergy, And allergy is a damaging immune response by the body. It is usually caused by pollen, fur, particular food, or dust, in which our body becomes hypersensitive. And when you're allergic to a certain thing, you have your rashes and sneezing, coughing. It affects. It really affects your well-being. There are some people who are so allergic that they could because an allergic reaction could also cause swelling of the various organs in the body, and when you have that, then sometimes you can no longer breathe, and some people really die from a strong allergic reaction. There are substances that induces an allergy, and these are referred to as allergen. And there are, allergens are found naturally in certain food. So I have here a list of the 14 food allergens. The cereals that contain gluten, wheat, for example, milk, tree nuts, celery, egg, soybean, sulfur dioxide, which is usually used in food, mollusk, fish, and so on. So we have 14 top food allergens. And there are many people who are very allergic to this food. And all they have to do actually is to be aware that you're allergic to a certain substance, and therefore you avoid that substance. We do not ban this food. So it's not a cause for banning of food, but it is, it is actually placed in food labels because, to avoid, to enable people to avoid exposure to these substances if they're allergic to it. On the other hand, there's also toxicity potential in certain foods. And this would, the toxicity would cause the loss of body functions or even death. And the potential depends on the type, the amount, and the susceptibility of the individual. Now, there are natural plant toxins that you can find in different plant products. And these toxins are species specific. You can only find that in particular species. And sometimes it is the substance that would give the plant its particular characteristics, like its color or its flavor. And the adverse health effects of toxins in plants usually range from an allergic reaction, severe stomach ache, vomiting, diarrhea, confusion, visual disturbances, salivation, hallucination, or even death. There could also be long-term health consequences if it could affect the immune system, the reproductive system, or the nervous system. There are also plant toxins that could cause cancer. So these are the natural toxins in food. There are 600 plant species that contain pyrrolicidin alkaloids. Okay, but these are usually present. These substances are usually present in very small amounts. Okay, and only when you start eating, probably in kilos, then you can suffer the consequence of that toxin. So even in these citrus fruits, you can find also a substance called furocoumarine, which is a natural toxin. Again, it's not present in large amount. Lectins in beans is also toxic when taken in large amount. And then you also have the glycoalkaloids in, they call this shade, nightshade plants, like in potato or in tomato, or even in eggplant. You also find a toxin in eggplant. But But again, these are not present in large amount. Or the cyanide compounds in bamboo shoots. That's why you have to slice the bamboo shoot in very thin slices and boil them so you can remove the cyanide compound there. And that's also true with cassava. You can remove that substance by boiling the cassava. Anti-nutrients are substances, that's another another substance that is not good for you, for your health, because these are substances that would block the absorption of certain nutrients. And of course, nutrients are the substances that would nourish our bodies so that we can grow and live. There are naturally found anti-nutrients in animals and many plant species. And these compounds are usually designed to protect the plants from infections or being eaten by insects. So I have here a list again of anti-nutrients. Glucosinolates are found in cruciferous vegetables, crucifers, broccoli, cabbage, bok choy. Sometimes, sometimes there are some people who are very sensitive to this and they're advised about not eating too much crucifers. So this prevents the absorption of iodine. And then lectins, these are found in legumes, beans, peanuts, soybeans, whole grains. And it can interfere with the absorption of minerals like calcium, iron, phosphorus. Oxalates are found in green leafy vegetables and tea. And this can bind the calcium and prevent it from being absorbed. You know, you need calcium, especially for the strengthening of bones. Phytic acid or phytates, it's a problem with us as well as with hogs, because what is that? It does is it decreases the absorption of minerals like magnesium and calcium. Saponin is another anti-nutrient. It's found in legumes and whole grains, so it can interfere with the natural absorption of nutrients. Tannins in tea. Okay, so this is tea, coffee, legumes, and it can decrease iron absorption. That's why you should not be drinking, you should not be taking your iron supplement with tea or coffee. Now, food and environmental safety are assessed in accordance or in harmonization with international standards. Codex Alimentarius has a food safety assessment of recombinant crops or recombinant foods like those derived from recombinant crops as well as from recombinant animals. And the Organization for Economic Development, Economic Cooperation and Development, or OECD, has also released a document made by its panel of experts also on food safety and environmental safety assessment. And then the Cartagena Protocol on Biosafety has the principles for risk assessment that that can be followed. The environmental safety issues with living organisms introduced into the environment are number one is the loss of biodiversity. That's why you have the Cartagena Protocol on Biosafety as part of the CBD, okay, because of the biodiversity issue. And the loss of biodiversity could be due to the invasiveness of the introduced species, because it reproduces rapidly, it competes with the food of the native fauna in the area, or it can be very aggressive also. And hybridization between introduced and native species is also would also result in the loss of biodiversity, and this has been found these have been found as problems already. And then, of course, the loss of biodiversity can also be caused by preference of farmers. They would prefer only to plant a certain variety, and then the other varieties would be lost. And the other issue that we found in the Philippines is the practice of farming in sloping areas. Now, invasiveness, here in the Philippines, the introduction of this water hyacinth. This is a native of South America. It was introduced as an ornamental plant in the Philippines. It has escaped in our water bodies, and it takes over the water body. It drains the water, changes the environment from free-flowing water to stagnant, shallow water. So it changes also not only the different plant species. Actually, there are few species that you can find within the area where you're going to find the water hyacinth. And then, of course, the associated insects and other animals would also be changed. So that because it becomes, the water hyacinth becomes a predominant vegetation, and it's a big problem actually, especially in marshy areas. On the other hand, there is also the hybridization between related species. It could lose the one pure individual species because you have genes that has been introduced by the new species. And then you can no longer find the original species. What you can find only are the hybrids. And then there's this extinction of populations or the species itself. Actually, one of the examples that we usually cite about the loss of one species because of the introduction of another species is in South Korea, where they lost the native rice species, and now only the rice species that is used for food that is found in South Korea. And there's many more cases that you can find both in plants and in animals. Well, this is a farming practice that we found here in the Philippines because corn can grow in grassy, in grassy areas. These areas have previously been grasslands, and our farmers found that they can grow this GM corn actually. Now the problem here is because when you grow this GM corn, it is herbicide tolerant, and you have a clean culture so that when water comes, then it simply flows through or under this corn so that it erodes the area. And that's why we have a program with the Department of Agriculture wherein we promote a system of hedgerows and contour farming, like what they practice with vegetables in the mountain provinces in the Philippines here, as you can see. It is the role of government to address the safety concerns of GMOs. And what they need to do is to establish a regulatory system. And usually the basis of decisions in that regulatory system are the results of the safety assessment or risk assessment. And we do the risk assessment prior to the commercial release of the product. So I would be very happy to listen or to answer questions that you have about food and environmental safety if you want to get in more detail. Thank you very much.
So, maraming salamat. Thank you very much, Dr. Nina. I have been listening to your presentations for several years, and I always learn new things with your presentation, ma'am. Salamat po. So, dear colleagues, I enjoin you to type in your questions at the Q&A box. So, while waiting for that, I'll read this first question. So, Ma'am Nina, what do you think is the better mode for a country? It legislate a entirely new biosafety law, or it use existing legislation?
Well, our experience in legislation is it takes 2 to 3 years or even never, so that if you legislate the regulation for modern biotechnology and the rapidity with which the field would change, it would be very difficult later if you have a law specific for the regulation of modern biotechnology to change it if there are new developments in modern biotechnology. And that's why in the Philippines, what we have is a regulatory framework that is based on existing laws. And the policy is released only by the executive branch. We do not have to go to the legislative branch to be able to make revisions, which we did actually. We have revised the national policy was first in the Philippines. The national policy on the regulation of modern biotech products was issued in 1990. And the coverage of that policy is mostly actually on the experiments or research on genetic engineering itself. So it is the process that is the basis for the regulation in there. Later, after the signing of the Cartagena Protocol on Biosafety, we changed our national policy to harmonize with the Cartagena Protocol on Biosafety. Again, that was quite easy. It took us only 6 months to be able to do that, to have that changed, and we had a new policy released in 2016. So that covers only the LMOs. And we're happy that we were able to do that because there are, again, the changes in modern biotech products like genome-edited products, which are similar to mutated products. And since we are not regulating mutated products, it is easier for us to avoid regulating gene-edited products, because we have already sufficient experience with mutated products. You know that in the world there are more than 3,000 varieties of crops that have been produced through mutation, induced mutation. So, and we do not have any problems with those. That's why we decided to simply focus our regulation only on LMOs as defined by the Cartagena Protocol on Biosafety.
Thank you, Dr. Nina. There's a question here from Nippon. I am Supasit. What is the environmental safety issues that needs to be assessed related to releasing GM crops?
Yeah, the loss of biodiversity is the issue in here, and the loss of biodiversity could be brought about by the hybridization between the introduced variety and the existing and related species, so that you could lose that native species. That's one of the issue that is being addressed. Another is when you have a GMO that has a pesticide that has an incorporated pesticide, the issue there is whether it will also adversely affect other organisms, because it adversely affects the target organism. So you have possible adverse effect on non-target organisms. So you have to study that. You have to assess whether that happens or not. The other is if you have an associated practice that could also have an adverse impact. Like in the Philippines, we used to plant corn only in level areas, not along sloping areas. But when our farmers found that they could plant corn in sloping areas, they started planting there without adopting the safeguards for to prevent soil and water, soil erosion, I should say. So that's also one of the issues that we have to address with our regulation.
Salamat, Dr. Nina.
Okay.
So there's this is not really a question, but just a comment from an anonymous person attendee. Just a comment. Hybridization could happen not only between GM and local species, but also between all species planted in the same area. Yes, correct. And a final question from Melinda Damle. I, I hope I pronounced the name correctly. What is the soil and irrigation water optimum pH? Electrical conductivity for the following crops. Optimum yield. Number 1, paddy rice. 2, wheat. 3, sorghum. 4, oilseed. Soybean, groundnut, sesame, mustard, cotton, sugarcane. The soil water drainage speed for above crops. In India, during waterlogging, drought and excess rain is experienced in the same year. In the same year, rainy season, monsoon. Do you think abiotic stress tolerant technology will help Indian farmers to overcome drought and waterlogging problems?
I think you have in India already this water submergence tolerant rice. It's according to the information that we got, is it helped the farmers very much that they planted during rainy season, and that the rice would the yield of the rice that has been submerged under flooding conditions has not been affected. We also have the same species, the same variety in the Philippines, a flood tolerant rice, but we don't have any figures yet on how much they would help actually with the farmers. So it's we have drought tolerant GM corn that has been introduced in the US, and they found that it helps very much during drought season. So actually, those are the things that we need for climate resilience. Actually, those are the traits that we need for climate resilience, and we're starting actually a program on those kind of crops.
Thank you, Dr. Nina. And just to clarify that the flood-tolerant crops Dr. Nina mentioned are not yet GM, but the drought-tolerant crops in the US is already GM. So if you still have questions for Dr. Nina, please type them over at the Q&A box, and Dr. Nina will try to answer them also through the Q&A box. But in the interest of time, we'll be proceeding to the next presenter. Ma'am Nina, maraming salamat ha. Thank you very
welcome,
much.
welcome.
So,
take care.
Salamat po. So let us now proceed with our next presenter. And he is a dear friend of mine. He is the Executive Director of the Philippine Seed Industry Association, or PSIA. Prior to that, he was the Senior Regulatory and Scientific Affairs Lead of Monsanto Philippines for 10 years. He also served as the Gene Bank Manager, the Director of Crop Biotech Center, and the Acting Deputy Executive Director for Research at the Philippine Rice Research Institute, or PhilRice. He is the local counterpart of IRRI. He holds a Master's of Philosophy in Plant Breeding from the University of Cambridge, UK, and a PhD in Genetics from the University of California, US. He is a recipient of awards for the 10 Outstanding Young Men of the Philippines, TOYM, very prestigious here in the Philippines. Outstanding Young Scientist from the National Academy of Science. And just last year, he was also awarded as one of the faces of biotechnology. And look at that face, dear colleagues, very handsome. So today, my dear friend Gabi will be sharing the road to commercialization of BT corn in the Philippines. So please welcome Dr. Gabriel Romero.
Hello, everyone. Thanks, Abe, for the introduction, and thanks to the organizers for inviting me. Let me now share my slides. This is my 10th year actually participating in the Farmers Exchange Program. And this time around, as a colleague from the PSIA. Our association is the largest and oldest seed association in the Philippines. We have 26 members from government, academe, dealers, Filipino-owned companies, and multinational companies. Some of our Filipino-owned companies are already having business outside the Philippines as well. So it's my pleasure to share with you a technological revolution that swept the Philippines in 2002, exactly 18 years ago. That was when the country started growing BT corn. You know, as soon as BT corn or the Mon A10 event was approved in the US in 1996, efforts to introduce it in the Philippines began in earnest. It's a GMO, hence there were concerns as to government policy, scientific expertise, facilities, and safety to humans, livestock, and non-target organisms, as mentioned by Dr. Halos earlier. Well, the technology was made first in the US to fight off the European strain of corn borer. But we have a different corn borer here in the Philippines. It's Asiatic corn borer. So the question is, will this technology also kill this devastating Asiatic corn borer in the Philippines? And needless to say, my friends, all these questions were answered, yes. We have the biosafety guidelines as early as 1991. And as we move along, there were new ones that were developed. There were also academics and government researchers, notably entomologists, molecular biologists, and geneticists, who only needed, some supplementary orientation on biotechnology. And there were seed companies who had experience in handling regulatory trials in the US. And we have this Institute of Plant Breeding in the Philippines, part of UPLB, that is a leading R&D organization. And there were also already world-class facilities available that only needed retrofitting for containment purposes. The evaluation was done in 3 stages. Greenhouse test to check if the BT corn was good against our local corn borer, and then followed by the confined field test and the multi-location field tests that were done to see if friendly and other non-target organisms were affected. For the greenhouse evaluation, 2 trials were done, both at IRI, the International Rice Research Institute in Los Baños, jointly by the seed companies and the IPB. Evaluation of the ACB damage data by the IBCs or the Institutional Biosafety Committees, and the NCBP, the National Committee on Biosafety of the Philippines, and the experts, the reviewers, they concluded that Mon A10 had excellent control against ACB. So yes, to the very basic question, then we can move to the next stage. Next phase was an open field trial, but a high fence to prevent pollen flow. Public approval was also necessary because there was a community involved, bigger community, unlike when it was done only in Erie. And this confined field test was conducted in Mindanao, so the southern part of the Philippines. Earlier, it was in Luzon. And the trial confirmed the efficacy and specificity against ACB. Other insects were not affected. And you know, farmers who were up against the trial saw the nice clean BT corn ears without ACB damage. And eventually, after the trial, they turned around to support the future trials, and they began demanding for seeds to use. So they were opponents, turned supporters, and eventually users. On to the third phase. Okay. But before proceeding to the third phase, we learned that during the confined field tests, that there were serious public concerns, and that was quite a surprise. The public seemed not ready for BT corn just because of the noise around the confined field trial. And so I think the stakeholders there, then at the time, the technology developers and the government intensified their outreach to farmers, consumers, the LGUs, just to counter the unfounded fear-mongering by certain anti-GM entities. And at the time, the precursor of BCP was already helping. It was a pro-biotech NGO that was already helping in doing the outreach, doing the briefing to other stakeholders. So the last regulatory trial was the field tests that were done in 19 locations around the country. So again, they needed public approvals, not just the technical approval from the DOST, from the scientists, but the local community, the villages, and the local executives had to give their consent for the trials. And again, the trial showed outstanding performance and specificity against ACB. And there was also a bonus. They observed that the diseases were also minimized in the BT corn. So with all these positive results from the 3 phases of local evaluation, plus the submitted documents on food and feed safety assessments done abroad, the Philippine regulators finally granted approval to BT corn MON 810, a landmark for Philippine agriculture. Okay. 6 years approval timeline for a controversial technology. This should be an amazing feat. That was made possible only because of a strong political will of the government, combined with sound policy, agile companies, and progressive scientists, and passionate farmers. And soon after the approval, we've seen the exponential growth of BT corn adoption from 2003 to 2010. And from 2011 to present, adoption is hovering the saturation point for yellow corn. There's still a lot of white corn to cover but for now, it's yellow corn. BT corn is mostly sold as a stock with herbicide tolerance, or HT. And the Philippines became a mega country for biotech in 2010 when it passed the 500,000 hectares mark. At present, I think we are 12 or 13 in the world among the mega biotech countries growing GM crops. There are 4 additional BT corn events approved in the country. There's another one from Monsanto, Mon 89034, 2 from Syngenta, and 1 from Pioneer. So there are now more choices that our farmers can use. Proper stewardship is necessary, especially now that the adoption is more than 80% of the yellow corn area. We need to prevent or delay the breakdown of BT corn control against ACB, and there are 2 ways to it. First is increasing the size of refuge or non-BT corn in the farm. And second, or in combination with this refuge strategy, is combining BT genes. With 2 or more BT genes, it will take many more generations of corn borer to overcome or become resistant to BT corn. A critical component of the insect resistance management is field monitoring. Everyone, all the stakeholders, especially the technology developers, the local agriculturists, municipal agriculturists, provincial agriculturists, and the farmers themselves, everyone should be on the lookout for resistance development. That means checking if these BT corn varieties are still effective against corn borer. And farmers are very happy with GM corn, primarily because of high yield, resistance to pests, and nice clean ears, very good quality. And academics have found concrete benefits of GM corn to farm productivity, farmers' income, and the environment. And I'd like to end by quoting 2 of the passionate and articulate farmer leaders we have in the Philippines, Rosalie Eliasus and Roger Navarro, who both grow BT corn themselves, and they vouch for its safety and profitability. We are proud of this BT corn experience, ladies and gentlemen, that we would like to share and help other countries in the region to enjoy this great technology as well. Good afternoon, and thanks to all of you.
Thank you for that very informative presentation, Gab. So please hold on to your questions. Again, everyone who would like to ask a question, type in at the Q&A or at the chat box, and we will be answering your questions after the last presentation. So our last but definitely not the least presenter for today's session is the president of the Australian Oilseeds Federation and a board member of the Cotton Research and Development Corporation. She is also the principal of the Baumann, Richards and Associate, a consulting firm specializing in providing strategic support to agribusiness firms in Australia. Our speaker has extensive experience in the biotechnology sector, including regulatory frameworks, protocol development for handling and marketing biotech crops, market analysis, and advocacy. She is currently involved in the commercialization of the super oleic, high oleic safflower, a unique bio-based feedstock. Ladies and gentlemen, to talk about Australia GM Canola and its road to commercialization, let us all welcome Dr. Rosemary Richards. Rosemary?
Thank you, and thank you very much to the organisers for the opportunity to speak at this forum, and it's been very informative to date. I'll just share my screen with you. So I'm going to talk to you a little bit about the road to commercialisation for GM canola in Australia. And compared to the relatively smooth path that our previous speaker talked about, you'll see that in Australia we had a bit more of a rocky road in terms of commercialisation of GM canola. But just to give you a bit of a flavour for GM crops in Australia, we actually have grown GM cotton for a very long time. So it was approved for commercial release in 1996, and today accounts for pretty much the whole of the cotton crop in Australia. And that is an insect and herbicide tolerance cotton. In terms of canola, as I said, the road to commercialisation was a little bit less smooth. And whilst we did approve, our regulator did approve canola for commercial release in 2003, it was actually not until 2008 that the first GM canola crops were grown in Australia, and that was only in 2 states. Western Australia came on board in 2010, and in a very positive move over the last couple of weeks, we've just seen South Australia allow GM canola. But because of that, and because of the delays that occurred with GM canola, it actually only accounts for 25% of our production today, even though Western Australia, where it is approved since 2010, is our largest canola producing state. And that all of the GM canola today that is grown is herbicide tolerance canola, but we also have approved a new omega-3 canola as well. And as was alluded to in the kind introduction, we also have a super high oleic safflower, which is a GM crop that's been approved for commercial release in 2019 and was grown for the first time last year. In terms of our framework, and our first speaker's done a very nice job in setting the scene for me here. In terms of our framework, we do have a national framework for gene technology regulation, and the focus of that national legislation is around health and safety of people and the environment. It doesn't include trade or marketing considerations. The Act is administered by our Office of Gene Technology Regulator, and that regulator is responsible for approving GM field crops and also commercial release of GM crops. The chart is a bit of a busy slide, I appreciate, and the chart at the top gives you a feel for the process that we go through in terms of approving GM crops. I guess just to highlight a couple of things. The risk assessment is very important, and our first speaker gave a very nice description of risk assessment, and we follow a very similar Codex compliant risk assessment framework. The other feature of our process is that there is quite a number of steps where both experts and the public can have input into that risk assessment and the decision-making, well, not so much a decision-making process, but certainly have the opportunity in the consultation process to put forward their views. And then we also have a look at what corrective actions should be taken in relation to any risks that are there, how might they be managed in a commercial sense, and then obviously the decision to approve or not approve is made. As I said, the GM canola pathway was a bit of a rocky road, and we did approve it in 2003. What happened was immediately after that, all canola growing states implemented moratoria that were based on what they called trade or marketing concerns. And essentially what happened then, there was a period of consultation with industry, consultation with consumers, consultation with our trading partners around the commercialisation of GM canola. And this did result in many years of delays and did reduce opportunities for farmers in those states. And it's probably also created a bit of an unclear pathway in terms of the future of GM crops in Australia. However, as I'll share in a few minutes, hopefully we've now got a bit of some improvement in that pathway now. And I think with some of the developments that's happened more recently with the approvals of the omega-3 canola and super high oleic safflower that are more consumer orientated products, I think we now have a smoother pathway for future GM crops. So this map just gives you a bit of a view of what happened with the states, and essentially it's a busy slide and I won't go into it in detail, but you can see essentially what happened was New South Wales, well, there's a fairly uneven landscape. So Queensland is happy to approve GM crops. There's no moratoria, and they've been growing GM cotton for a long time. Obviously, there isn't any GM canola in our northern Australia. New South Wales and Victoria implemented moratoria. Today, Victoria doesn't have any constraints. New South Wales still has legislation around food crops, and so there is, if GM food crop comes to market, they can it still needs to go through a New South Wales approval process. South Australia was the one that was holding out for quite some time, and just over the last 12 months or so, there's been considerable work, number of reviews undertaken in South Australia. And just as I said, in the last couple of weeks, South Australia has now approved GM to be grown in all of the mainland part of South Australia. There is one island off South Australia, Kangaroo Island, where it's still GM free. And Tasmania remains a GM free state. So essentially today you can say that Australia mainland has approved GM canola, whilst our island states have not yet approved GM canola. So what really occurred then was that industry, the process that we took to move people forward on the GM canola discussion when the states put in place the moratoria, was really the industry said we need to be able to demonstrate a market choice approach. So recognising that some of our important trading partners didn't want, were happy to continue to buy non-GM canola. We said that the industry put in place a market choice approach. And the Australian Oilseeds Federation was a leader in terms of trying to pull the industry together to develop a framework for market choice. So we did develop this industry framework to manage approved GM varieties. And then the focus of that was to ensure that we can not only provide confidence to the consumers and to industry and to government, but also minimise any disruption to trade that might occur from trading both GM and non-GM products. Our framework has three core components. Obviously, first of all is the Australian approvals. So we need to, any product needs to go through the Australian approval process. Before that process gets what we call industry freedom to operate, it needs to go through a market risk assessment and obtain approvals in what the industry believes are the key export markets, those key markets that we would trade with. And then there also needs to be a market risk management strategy. So essentially that's the stewardship part. So the industry commits to putting in place a stewardship to manage GM and non-GM in a coexistence way. And we have a number of systems and protocols that can meet all of our customer needs. And the picture on the left is the document. It's downloadable from the Australian Oilseeds Federation website. And I would encourage you, if you're looking at a marketplace to deliver choice, it's a very useful framework to look at. It's the framework supported by the Australian grain industry supply chain management process. Australia is the only exporting country in the world to have a code of practice or code of conduct that all of the grain industry and all the exporters sign up to, and that's the document there on the bottom right hand corner. And it's a very powerful tool that we use to again provide confidence to our customers that we have the systems to support it. We sort of look at as GM as a quality parameter, and that is really managed by industry, not by government. So a government approves GM crops for commercial production, and once they come through that approval process, then it's really an industry management approach to ensure that we can deliver that market choice. We have a series of information management systems in place to identify market requirements, develop standards, and then we support that through the Grain Trade Australia contracts and trade rules that support that. And we have a series of industry processes, common processes that have been developed to support, so technical guideline documents, procedure documents, again, to support that confidence. And the two core documents that the industry works to is the top one there is the grower. Growing Australian grain is the code of practice for the growing or production sector, and the document on the bottom is the one for the supply chain or the post-farm gate system. So in summary, I think one of the things that Australia does have is a very effective and respected regulatory framework. I think our framework is our Office of Gene Technology Regulator, supported by our Food Standards organisation, have done a lot of, are very highly respected around the world, and that certainly gives our industry a lot of confidence that when we do have GM crops approved, that certainly all of the safety checks for human and environmental safety have been appropriately undertaken. As I said, one of the complications that we have in Australia is that our states have the responsibilities in relation to trade and market considerations. And in the early days of GM, particularly GM Canola, which was our first GM food crop to come into production in Australia, the state responsibility or the state reactions certainly caused some challenges for the industry in terms of moving that GM crop through to commercialisation. But I think we have now, with some of the recent developments that we've seen, particularly in South Australia, I think that's moving forward in a very positive way. But what it did do is result in an inconsistent approach to the commercialisation of GM crops. And again, as I said, we're seeking alignment around that. The industry's market choice approach has been a very positive tool for us, and certainly now since 2008, Australia has successfully traded non-GM and GM canola from Australia around the world, and we haven't experienced any difficulties in delivering against the promise of that market choice program. And as I mentioned, we are now starting to see some newer consumer-focused products coming through, the omega-3 canola, and in the one I'm involved with, the super high oleic safflower, which is an industrial oil product. We've certainly seen a much smoother path to market for those consumer-focused products, and hopefully that will be the way of the future. So hopefully we've learned some lessons from the GM canola experience, and we can move forward with a much smoother pathway to market for some of our newer products that are coming through the supply chain. So hopefully that's given you a quick overview of the Australian regulatory framework, and I'm very happy to answer any questions that you might have. Thank you.
Thank you very much. Thank you very much, Rosemary. I'm sure, like myself, our participants benefited very much with your presentation, not only on GM canola, but with the Australian regulatory process for GM crops. So we'll now open the floor to your questions for our two last speakers. And I invite now Rosemary and Gabby. So Rosemary and Gabby, that sounds like a movie title. So Rosemary and Gabby, Thank you for those nice presentations. One is a smooth path, one is a rocky road, but it all went into a successful GM crop introduction. The first question, Gabi, is for you. This is from Nippon Iam Supasit. Have you find any insect resistance to BT corn since its introduction?
Well, there is continuous monitoring of insect resistance development, and that is being done in cooperation with scientists in the university, with academics. There is continuous collection of larvae from different corn areas in the Philippines. Well, so far, there's no resistance development found in the field. There are some suggestions, but there are some larvae that survive on BT corn plants, but they do not survive when they are mated to susceptible strains. The offspring do not survive feeding on BT corn. So the short answer is none so far, but there should be close monitoring because there are some suggestions but not confirmed.
Thank you, Gab. So our second question is addressed to Rosemary. Rosemary, Australia is known very well for its science-based but stringent regulatory process. If that is the case, why is it that some states, for example, South Australia and Tasmania, are relatively anti-GM compared to the rest of the country?
Yeah, thank you for the question. I think, yeah, we do have a very stringent system, and as I say, we do have a high, there is a high respect for our regulators in Australia. South Australia and Tasmania both have very highly valued food industries. So they've built a marketing platform around their wine industries and their food industries, and I think they saw in the early days that there was potential to for consumers to perceive that if there was GM crops grown, that you know, that sort of very clean, green and specialty food image would go. And that's one of the things that we've done very successfully with South Australia, and you know, the grain producers organisation in South Australia has done a very good job in educating and communicating to the wider industry and engaging the food sector over there to show that even though there's GM crops being grown in the other states, like New South Wales and Victoria, that it wasn't damaging the food brands in those states, and those states still had very strong image for the quality of their food, even though they were growing GM crops. And I think that's what's moved South Australia now to allow GM canola to be grown, is that the food industry has realised that the two can coexist. So I think it's just it's an education and awareness thing. But for Tasmania, it's very much around that, you know, they have that clean green image, and yeah, they like, you know, at the moment it's something that they don't see that the benefits of, and they're not a big canola growing state, they're not a big grain growing state. They don't see the benefits would outweigh that issue around food marketing for them.
Okay.
Thank you.
Thank you, Rosemary. So thank you for enlightening us on the idiosyncrasies that exist in different states, even if the national government has this stringent science-based regulation. Gabi, this second question is from Abison. It's related to the first question. Is there any monitor on the changes of occurrence of insects after BT corn is commercialized? For example, major insects and minor insects?
Yeah, the target pests and the other insects are continuously monitored there is these so-called sentinel sites that are being regularly set up and then monitored by technology developers in partnership with the BPI and the NCPC, Bureau of Plant Industry, and the National Crop Protection Center. So these are always monitored, and there's a long data sheet that has to be filled out by technology developers. Yes, there is the 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 incidence of these major insects and the friendly insects uh, is continuously monitored in the field. And so far, we've not seen dramatic changes as to the population. The good thing is the friendly insects continue to abound, and the target pest, the corn borer, the the pressure is continuously low throughout these years since the BT corn introduction.
Thank you, Gab. So it's really targeted against the harmful insects. I hope we have also this targeted technology against insects in Malacanang. So our next question is for Rosemary. Hi, Rosemary. Do you have anti-GM groups also in Australia? If so, how do you cope with them?
Yes, we do. I think, like everywhere around the world, there's people across the spectrum in relation to GM crops. Look, back in the sort of when GM canola was approved back in 2003, there was a very strong anti-GM group, and I guess that was reflected around the world as well. Today, that group exists, but I think it's much smaller than it used to be. I think people are much more comfortable now with the technology, and there's been other issues, food allergens and other things that have come across consumers that are perhaps more concerning. So we do still have a anti-GM movement in Australia, and in some respects our consumers are quite aligned with European consumers in some of their the way that they think about these things. But I think what we've done as an industry is that we've consistently just been very transparent about reporting about the progress that we've made, and as I said, we've successfully exported many millions of tons of canola over the last decade or more without any problems. And so I think the concerns that the anti-group was raising around what might happen as a result of GM canola, as the industry we've been able to be very transparent and able to provide the evidence to show that those concerns haven't arisen, and therefore I guess the anti-GM movement has not been able to get as much air in recent times as it has previously. So no, it looks still there, but I think it's a way to work with these groups, and right back at the very beginning, we involved the organic groups and all of the people in our supply chain. We involved them in our discussions and the development of our market choice framework, so that they felt part of that, and hopefully that gave confidence that the frameworks and the systems that we have in place can help allay their concerns.
Thank you, Rosemary. Same here in the Philippines, so thank you very much. So last two questions. The first question is to Gab. Gab, please, very short answer. And this comes from your colleague from Syngenta. Why is it corn is the only GM product that has been commercialized in the Philippines? And this comes from Ms. Jenny Panopio.
Oh, I think she's from Corteva. Okay. Anyway, well, the GM experience in the Philippines is not as smooth as I incorrectly portrayed. There were also bumps, and one major bump was, the nullification of our GM policy I think in 2015. But anyway, we already got a new GM policy in 2016. But anyway, there's a lot of regulatory, I would say, bureaucracy also still existing. That's why the other GM crops have not seen approval or propagation in the Philippines. But I know there are others in the pipeline, like BT eggplant already nearing approval, BT cotton, and golden rice, they're already nearing approval. I think because some of the hurdles or some of the kinks in the regulatory process are now being ironed out. So sooner or later, we will see more new products in the market. Thank you.
Thank you. And the last question for Rosemary. Short response, Rosemary. This is from the land of India. The question is there the R&D or the R&D activities in Australia, are they being provided also to are they being provided also to any Indian organization? Do you have tie-ups, Rosemary?
Yes,
Are
so
you
we.
aware?
Thank you for the question. Australia has a lot of collaborations around the world, and very much so with India and with China and with a number of other countries. In terms of specifics, I need to take the question on notice, but certainly through our Grains Research and Development Corporation, there is a lot of participation in international initiatives, and I know there is quite a bit of work in the canola program through GRDC. That's in cooperation with India and also across our pulse crops as well. So yeah, look, we certainly have a lot of engagement at the R&D level with India, with some of our other trading partners, and yeah, look, I'm happy to provide a link to the GRDC that you could find more details on the specifics of that collaboration. Thanks.
Thank you, Rosemary. So there are other questions. Unfortunately, in the interest of time, and we would like to give away some major prizes. Please, Gabby and Rosemary, there are questions here at the chat box and at the Q&A box. If you could answer these off-site, particularly those from our colleagues from Debarati Das, from Paulo Veluz, and also again from Milanda Damle. So with that, I am now closing, at least online, the Q&A portion of our webinar. And ladies and gentlemen, please help me again thank from the bottom of our hearts our two great speakers, Rosemary and Gabby. Thank you very much. Maraming salamat po. Maraming salamat. Thank you also to all who participated today. I would like to invite you for tomorrow's third day webinar where they will be discussing biotech and science communication. This is again a very interesting topic. So with that, everyone, I will be closing today's session. Thank you for attending the second day of the 14th Pan-Asia Farmers Exchange Program. Salamat and mabuhay.